Our next speaker is Dr. Robin Hopkins, and she received her bachelor's degree from Brown University, and she double majored in biology and gender studies. And she went on to do her PhD at Duke University with Dr. Mark Rauscher, and she studied flower color and its role in speciation. She then received an NSF postdoctoral fellow and fellowship and she went to the University of Texas at Austin and worked with Dr. Mark Kirkpatrick. And in 2014, she started her lab at Harvard University in the Department of Organismal and Evolutionary Biology. And she has received several awards, two of which, one is the New Phytologist Award, Tansley Medal, and also the American Society of Naturalists Jasper J. Loftus Hill's Young Investigator Award. So congratulations. She's published many papers. Some journals include New Phytologist, American Naturalist, Science, Evolution, and Nature. And please join me in welcoming her today.
Awesome, and again, thank you to the organizers for inviting me and organizing this. It's really an amazing opportunity to get to think outside my little box about bigger and broader questions, and thank you all for coming. So I sort of fit in the other end of the scale. I think about microevolutionary processes of coexistence and speciation. So in particular, do I even have the right? Ooh, I think so. There's a lot of them around here. Let's see. No? Nope, that one's doing that one. Thank you. Technology. All right. So in my lab, and what I'm interested in studying are these processes that influence speciation. So how do we get from one interbreeding group of organisms to two distinct groups of organisms that no longer interbreed and then are allowed to accumulate differences phenotypically and genetically? And so conceptually, we know that there are lots of processes that influence speciation and the accumulation of reproductive isolation. And these processes range from natural selection, sexual selection, genomic conflict, drift, and of course, just geography. But my work focuses specifically on selection and the role of selection in the evolution of reproductive isolation. And in particular, the role of natural selection in sympatric populations in driving reproductive isolation. So I've studied this process called reinforcement. And by definition, it is an increase in reproductive isolation due to selection to decrease hybridization or mating. That's often very wordy and hard to get your brain around, so I'm going to describe it in terms of a scenario. So we have divergence, often in Allopatry, where two diverging taxa start to accumulate differences. Now, if these taxa or incipient species come into secondary contact in sympatry in the same geographic area, they might produce hybrids. And if those hybrids are maladapted, partially sterile, maybe inviable, a number of things can happen. We could get local extinction. Hybridization is costly. One species hybridizes too much and goes away, disappears, goes extinct. We could get fusion of the two taxa such that any differences that were accumulated in Allopatry are now swamped out through gene flow. Or we could get divergence to stop this hybridization. And that divergence, the evolution of those characters that increase reproductive isolation, is the process of reinforcement. So in this case, one of these flowering plants evolves red flowers, attracts a new pollinator. This is favored simply because now these species are no longer exchanging pollen and therefore not creating these maladapted hybrids. So this process of reinforcement is often characterized by this pattern of greater reproductive isolation in sympatric populations than allopatric populations. And so we get the divergence of characters in sympatry to increase reproductive isolation. That pattern has been observed across the tree of life. So we see it in plants, in insects, in birds, in mammals, in all sorts of traits from mating song calls to chemical pheromones to behavioral differences, flowering time, where we see that there's this presumed increase in reproductive isolation in sympatric populations relative to what we see in allopatric populations. So this diversity of patterns across the tree of life implies that this process of reinforcement might be really important for the process of speciation and for the coexistence of these closely related taxa, or maybe even formation of these species. But I would argue that we know very little about this process of how it happens and why it happens. Particularly, what are the genes involved in these traits that are diverging? How much reproductive isolation does each mutation cause? What is the strength of selection that actually drives this divergence in sympatric populations, allowing reinforcement to occur instead of extinction or hybridization and fusion? And then what are the mechanisms of selection? So I'm going to present a case study of this process of reinforcement in my favorite annual wildflower, which is Phloxera mundii. So this is a Texas wildflower. It blooms in the spring along with a huge bouquet of other flowers. It's pollinated by butterflies, mostly this pipevine swallowtail, Batis philonor. And it comes in two flower color varieties. So this violet flower color that I'm going to call light blue, and this dark red flower color. And what has been very interesting about this species is the geographic distribution of this flower color polymorphism. So most populations have light blue individuals. Central part of its range, they're all light blue. But in the eastern populations, all of the individuals have these dark red flower colors. And it's in this portion of the range that we have sympatry with a closely related species, Phlox cuspidata. And this second species of phlox has that same light blue flower color. So in the 80s, Don Levin observed this pattern and hypothesized that this divergence of flower color in sympatry decreased hybridization and was a result of reinforcement. So I wanted to know, okay, what's the genetic basis of that trait of flower color differences? And do any of those genetic mutations actually decrease hybridization? And what is the strength of selection that drove this change in flower color in sympatry? And lastly, how does this work? What is the mechanism of selection? So the first question, what is the genetic basis of this trait? Again, this is a polymorphism within species. So I can use classic genetic crosses to start to get at the genetic architecture of this flower color difference. I cross the light blue variety with the dark red variety. I get an F1, I self it, I get F2s, and I get 4 flower colors. I regain my parents, light blue and dark red. I get my F1 color, dark blue, and this novel light red flower color. And I got these flower colors in a beautiful 9:3 to 3:1 ratio that all intro bio students will tell you means there's 2 genes with complete dominance. One locus corresponds to flower color hue, and this is the type of anthocyanin pigments produced in the flower. So these blue guys produce blue and red pigments, and these red guys only produce red pigments. The intensity locus, the second locus, controls the amount of pigment produced. So the ancestral morphology is light, such that you'd produce a little bit of pigment, and the derived mutation causes a 3-fold increase in the amount of pigment produced in the flower. So these pigments are anthocyanin pigments, and anthocyanins are produced in all sorts of plants, and the biochemical pathway necessary to create these pigments is known. So we know the genes involved in making these pigments and can use a candidate gene approach to determine what the actual genes are, changes are in the genes causing this flower color difference. So using association studies in my F2 population, I found a perfect genetic association with 2 genes in the anthocyanin biosynthesis pathway. So here's my pathway, here are my pigments that are produced, and one gene, F3'5'H, perfectly segregates, variants in that gene perfectly segregate with flower color hue. So this enzyme changes the precursor of red pigments into blue pigments, and a loss of expression of that enzyme creates red flower color. The second gene is a transcription factor. It's an R2-R3 MIB transcription factor. And an upregulation of that transcription factor causes an upregulation of all of these genes in the anthocyanin pathway, resulting in a whole lot more pigment in the flowers. So what we found was that there's just two genes involved in this flower color change. One affects the type of pigments produced, one affects the amount of pigment produced. Now, of course, we go back to the maybe interesting biology. What do either or both of these mutations have to do with reproductive isolation? And is Don Levin's hypothesis correct that they're evolved because of reinforcement? So again, we go to this area of sympatry, and we want to know, do dark red flowers actually hybridize less than light blue flowers? And is that because they're dark or because they're red? So to look at this, I did a common garden field experiment in the sympatric range, where I grew all 4 of the possible Phlox drumundii flower colors in the presence of that second species, Phlox cuspidata. I allowed them to grow naturally, pollinators come naturally, set seed naturally. I then collected the seeds from the drumundii plants, grew them up in the greenhouse, and genotyped those offspring to determine if the dad was drumundii or the dad was cuspidata. And in this way, I could calculate the hybridization rate for light blue, light red, and dark red, and see which mutation, if either, decreased hybridization. Okay, so first you probably want to know that these species actually hybridize. And they do, a lot. Okay, so turns out 40% of the offspring of light blue drumundii individuals were hybrids. Now, there were reports in the literature that these hybrids are highly sterile. So that's a bummer for a mom creating a hybrid offspring, right? There's strong selection against doing that. So there's a potential for strong selection. Hybridization happens. Do dark individuals hybridize less, or do red individuals hybridize less? Turns out dark individuals hybridize a whole lot less than light individuals. So looking at the hybridization rates of dark blue and dark red Phlox drumundii, we find they hybridize at about 15 to 20%. But light blue and light red hybridize at 35 to 40%. So a change in a single gene decreases hybridization by 50%. You change your color, and you stop hybridizing, at least mostly. What's happening at the hue locus? Everybody asks me, and I have no idea. So we saw no signal of decreased hybridization when you're red versus blue. It was only if you're dark versus light, which is interesting and maybe gives me hope for the rest of my career. I have something to find. I don't know. I don't know how to figure it out, but I don't have any answers. But at least one of the mutations does decrease hybridization. And so it does look like there's a possibility that these traits evolved, at least part of flower color evolved in order to decrease hybridization in that sympatric population, and therefore prevent either extinction or fusion of the two species. So that's reproductive isolation. But what about the strength of selection? Right? So how did this trait evolve over time under selection? First, we want to know what determines the strength of selection when a trait is evolving due to reinforcement. Well, there's 2 factors that we have to consider. The first is the cost of hybrid mating. That's where the cost comes from, the reduction in fitness. And the second is the frequency of hybrid mating. So we can look at the cost of hybrid mating pretty easily. We can cross these 2 species in the lab and greenhouses and see how their hybrids do and whether or not they're sterile. So a postdoc of mine is systematically doing this, and basically these hybrids survive just fine. They look healthy and seem to germinate just dandy, but they're very sterile. So we're looking at two different types of crosses. We have Cuspidata as the mom and Drummondii as the dad, and Drummondii as the mom and Cuspidata as the dad. And you're looking at sterility of those hybrid offsprings. Turns out when Drummondii is the mom, these hybrid offspring are completely sterile. All pollen is inviable, and no seeds will set no matter what you cross it to. When the hybrids have Cuspidata as the mom, you can get some backcross and even interhybrid matings to set seed, and some pollen is viable. But sterility is still very high, almost between 65 and 70%, 75%. So this implies the cost of hybrid mating is super high, right? You definitely don't want to make hybrids. But the other part of understanding the strength of reinforcement or reinforcing selection is the frequency of hybrid matings. So I could measure that in my common garden field experiment. I planted those plants, I could calculate the frequency of hybridization, and I can calculate selection. So in that common garden field experiment, we estimated strong selection favoring the dark allele. But what does that mean about the strength of selection happening in Texas, right? These frequencies of the two species vary over space and over time, implying that the hybridization rate is going to vary over space and time. And so you could go out to every single population, or as many as you could find, and determine the hybridization rates. And then, since you know the cost of hybrids, estimate the strength of selection. But that's pretty hard. So what I did in collaboration with my postdoc advisor was to take a Klein theory approach. So the change in flower color occurs over a very short geographic space. And it's a very sharp change in frequency, a very sharp Klein in flower color. And we know from basic Klein theory that the shape of that Klein is determined by the strength of selection and the strength of migration. So the stronger the selection, the sharper the Klein, the stronger migration, the weaker the Klein. Right? So these are two opposing forces that can control the shape of the Klein. So what does flower color Klein look like? And can we use this idea of Klein theory to estimate the strength of selection that causes dark red flower color to be fixed in sympatry across this small geographic space? So what you're looking at is the Klein in flower color across one transect, where over here on these western populations, we have mostly light blue flocks, shown as these light blue part of the pie diagram. And over here, all the flocks, Drummondii, are dark red. And this change, and in the middle, you also see these light red and dark blue flower colors. And this change in phenotypic frequency happens over about 7 kilometers. So if you're in shape, you can walk it really easily and just see this flower color change over space. I'm also going to show you these Kleins like this, where we're just looking at the frequency of a phenotype across distance, where again we start with a lot of light blue, and we end up with a lot of dark red as we move from west to east. So I took a 3-step approach. First, I quantified the Klein, observing populations across this natural range in Texas. Second, I created a spatially explicit model based on the geography and the genetics of the flock system. And then I fit the model to the data in order to estimate selection and migration. And again, this work was done with my postdoc advisor, Mark Kirkpatrick, and a graduate student in his lab at the time, Rafael Guerrero. Oops. Step 1, the data. I think the fun part. We went to 39 populations across 5 transects and phenotyped over 12,000 individuals for flower color. So this was all based on the phenotype of the plants, not the genotype in this case. And you can see the 5 clines in these pie charts where we go from light blue to dark red. And in each case, we change the flower color in about 7 kilometers. So all of these clines were super sharp. And then we created this population genetic model to try to describe this system. So this involved modeling transects of phlox populations. Each population was a half a kilometer apart. Within each population, you can imagine a phlox doing reproduction, undergoing selection, and migrating within the population, and obviously migrating between populations. So the parameters in my model included, this transect was divided into 2 zones. You can imagine Allopatry and Sympatry, or where you should be light blue and where you should be dark red. So the first 5 parameters in the model were the location of that border. And these are sort of like nuisance parameters in the model, so we're just trying to fit the 5 modeled transects to the 5 observed transects and make sure they all line up properly. So that's the location of the border. Of course, we're interested in estimating selection, and so we had 6 fitness parameters in the model. So we have zones of Allopatry, zone of Sympatry, and 4 flower colors within each zone. One of them had a relative fitness set to 1, and we chose the most frequent flower color just for ease in the model. So light blue was 1 in Allopatry, dark red was 1 in Sympatry. And then the relative fitness of the other 3 flower colors were parameters in the model. And of course, there's migration between all of these populations. And so we used a symmetrical geometric dispersal function, which basically allowed a lot of migration to the nearest neighbor, a little less to the next closest neighbor, and then a little less as you get more and more distant. So in total, we had 12 parameters in our model. And I could put just about any value I wanted into these parameters and allow these populations of phlox to reproduce, undergo selection, undergo migration, generation after generation, until they reached equilibrium. And then at equilibrium, my model spits out the phenotypic frequencies of flower color across the transects. So I start with any parameter value I want, and I end with the predicted phenotypic frequencies across space. Then I take the observed data that I collected in the field, and I say, all right, what fits, right? Does the model output look at all like what I observed in nature? And sometimes it didn't, right? I still get light blue and dark red in different ends of the transect, but the shape of that curve was way wrong. And other times it was a really good fit, right? Where we see the lines going right through the observed points. The observed points are the boxes here. So I use likelihood to fit the model and data, and I search parameter space using Markov chain Monte Carlo, which gave me parameter estimates and confidence intervals around those estimates. So in the interest of time, I'm just going to show you what the strength of selection is in Sympatry that can give you that sharp climb. So here's the best fit model, first of all, where we align all of the borders in all 5 of the transects. And we can see that allele frequencies or phenotypic frequencies change pretty drastically over about 7 kilometers, going from all light blue to all dark red. And in Sympatry, the strength of selection is really strong. So in order to get this sharp climb pattern, it requires super strong selection acting against that light blue phenotype. In fact, these light blue Phlox stromundii plants suffer an 83% loss of fitness. That's incredibly strong. So this implies, based on the field studies that we did, we believe that selection, at least in part, is driven by reinforcement, this need to decrease hybridization. And based on this Klein model, we can predict that the strength of that selection is really strong. So we know hybridization is costly. This implies that at the time of evolution and since then the threat of hybridization was pretty frequent, and therefore causing strong selection favoring any trait that decreases that hybridization. So we have very strong selection in Sympatry. I'm not going to show this, but this model also confirms that there has to be, and logically there should be, weak selection in Allopatry maintaining that light blue phenotype. So we have this balance of selection forces that can maintain that sharp climb across geographic space. So now, what I think is super interesting and a huge future direction of my lab is understanding what the mechanism of that selection is. How do we get a decrease in hybridization, and how do we have this super strong selection favoring dark red flower color? And for that, we turn to the pollinators. So I mentioned in the beginning that these plants are pollinated by butterflies. In fact, both Phlox drummondii and Phlox cuspidata and all of the colors of drummondii are pollinated by the same butterflies. Most of the visits are from Batis philanor, this pipevine swallowtail. In fact, upwards of 75% of the visits are from this butterfly. But there's also a number of skipper species in Texas that will visit both cuspidata and drummondii. So in just initial observations, I could see that both species were visiting, both butterflies were visiting both species of Phlox. And so what I wanted to do is look more carefully at how they were visiting the different species and how they were moving between or within species based on flower color, and if this could cause reproductive isolation. So to do that, I set up these big potted plant arrays of Phlox. So I took drummondii plants and cuspidata plants, and I alternated them in these big arrays. And with the drummondii plants, I actually took two different colors. So I took that ancestral light blue flower color and one of the derived colors. And then I watched as the pollinators in the region came and visited the arrays, and I saw how they moved between and within species in the different color arrays. So I had three array types, one looking at the effect of drummondii flower hue on pollinator movement. So that's including these light blue drummondii and the light red drummondii alternated with cuspidata. And a second array looking at flower color intensity and its effect on pollinator movement. And finally, looking at the effect of both change in hue and flower color intensity on the way pollinators move between the two species of Phlox. So I'm going to present these data like this, where I have the transition percentages between species. So a pollinator that was on a light blue drummondii plant, 28% of the time, the next plant it visited was a cuspidata plant. So that's moving pollen between species. And for a light blue drummondii, 24% of the time, a pollinator moved to then a cuspidata plant. So hopefully you can see that flower color hue is not changing the transition percentages for these butterflies, right? Doesn't matter if you're light blue or light red, you still are moving between drummondii and cuspidata. And the same is true in the other direction. So if a pollinator is on cuspidata, it moves to light blue and it moves to dark red just fine all the time. But when we look at flower color intensity, we see a very different story. So in this case, pollinators are not moving between dark red drummondii and cuspidata, at least not nearly as much as they move between light blue drummondii and cuspidata. And so we see that this change in flower color intensity changes how the pollinators are moving between the species, and therefore how the pollinators are moving pollen between the species. We see the same pattern when we look at the array that included the dark red drummondii and the light blue drummondii. Again, pollinators are just not moving between drummondii and cuspidata when drummondii has these dark red flower colors. So importantly, so what we think the behavior that is going on is this individual specialization by pollinators, where a given individual of Batis, for example, goes from dark red to dark red to dark red to dark red. And another individual of that species will go from light blue to light blue to light blue to light blue. And when it does that, it doesn't differentiate between Drummondii and Cuspidata. And so it's moving pollen between the species. But that pollinator that goes to dark red is not going to Cuspidata because it's only going to dark red flowers. So this individual level specialization on color is allowing these plants to then be reproductively isolated. And importantly, this behavior that we see corresponds with the hybridization results that we got in the field, such that when the pollinators are not moving between species because the flowers are dark, we're also getting much lower hybridization rates in this field experiment, where we didn't look at pollinator movement at all. So I would argue in this case, the mechanism of selection involves pollinator behavior, or yet another species interacting in this system, where we get a 50% reduction in hybridization simply because of the way pollinators move between species differentially depending on the color of that species. So we start with a simple within-species polymorphism, right? The species has two different flower colors. We then realize that the reason that species changed its flower color or evolved a new flower color is because it existed in sympatry with a second species, and that hybridization is costly and therefore favors a trait that decreases hybridization. So we get the evolution of a polymorphism within species driven by the selection due to hybridization of a second species. And then we find out that a third species in that community is actually the mechanism through which the selection can act. So it's the pollinator behavior that allows this flower color change to actually result in decreased hybridization. So this may have a single genetic basis, and that allowed us to break up the different components of flower color and understand where selection is acting and how reproductive isolation evolved. And there's clearly at least one of the mutations is decreasing reproductive isolation, and we don't know what the other one is doing yet. And we know based on this model that there's really strong selection driving this flower color change in this system, presumably due to reinforcing selection. And we also have realized that it's this pollinator behavior, the behavior of this other species in this Texas, that is actually allowing this polymorphism to decrease hybridization. So with that, I'll take any questions and give thanks to my collaborators and mentors over the years. Thank you. Uh-huh.
The color.
Uh-huh.
Are they quote unquote seeing the colors so much, or do you think it's perhaps just the intensity of the colors so much is what they're actually seeing?
So yeah, the question was about pollinator vision and whether they can see the colors or if it's something else about the flowers that's different. So we think, so I can speak for the Batis, the pipevine swallowtail, much more than the skippers. We know a little less about them. So at least in Batis, controlled experiments have shown that they can tell the difference between blue and red, and they behave based on those differences. Other experiments that we've done show that they can show preference for blue over red. So they can behave based on that, even in this flock system. So they can see it, but they don't seem to be acting on it in this case. The intensity increases pigment around the whole flower, but in particular, you lose that white star in the center. So one hypothesis is that it might actually be that white star that is the difference, the white versus the dark star. And so we're currently starting to do experiments with a colony of Batis we're rearing in the lab to start pulling apart what is responsible for the behaviors that we see. So I think that's a good question. We don't have the answer. Great questions. So first, what's the cost of being red? It turns out that in Allopatry, there's a pretty clear signal that pollinators prefer light blue over dark red. So there seems to be in Batis, also based on some controlled lab experiments that batis have an innate preference for blue over red. And whether the innate preference or something learned is responsible for it, we definitely see in that allopatric zone that pollinators prefer light blue over dark red. And so we think that there's a paternal fitness advantage such that light blue pollen is moved more readily than dark red pollen. And so I think that's what's responsible for keeping that sharp climb. In terms of, so the second part of the question was, is Drummondii, dark red Drummondii taking over or spreading into the range of Cuspidata, that other species? And I don't have a good answer to that, but I do think that the actual range limits for these species is not so dependent on the co-occurrence of each other, right? And the reason I say this is that there's actually 3 Texas phlox species in its little clade. There's Cuspidata, Drummondii, and then there's a third one, Ramariana. And each of them actually specialize on slightly different soil types. And so I think that there's abiotic factors that each species is locally adapted to, and that there's regions in the range where they both can co-occur, but probably neither of them is really optimal. So I think that there's other things limiting the range of Drummondii on that eastern edge. I don't have any real evidence for that, but that's sort of the perception I have, yeah.
I'm just curious about the transactional nature of the pollinator and the flower. So is there any evidence for, and there is a sympatry for a difference in nectar or the reward given to the pollinator?
That's a great question. So we've started to look at nectar in these species, and like many butterfly pollinated plants, there's just very little bits of nectar. But based on our initial quantifications in the lab, there's maybe a slight difference in nectar amount such that light blue make a little bit more, but we don't see any difference in the field. And so we're looking into that more and trying to figure out if pollinators prefer light blue because there is indeed a better reward. So we know between species there's a huge difference in nectar reward, where Cuspidata is a selfing species, self-compatible and has high selfing rates. And like many selfing plants, it has a decreased showy flower display and a decreased reward. So there's definitely reasons in Sympatry to go to dark red Drummondii, but we're not sure what the implications of that are in the behavior right now. Yeah.
The hybrid incompatibility itself be an emergent trait. Have you tried crossing individuals that are far from that zone of Sympatry?
Yeah, so we did those crosses. I didn't show you the variance here on the incompatibilities. There's huge variance such that hybrid sterility can be really high or really low depending on the cross. And we don't see a geographic pattern that explains that variation. We're still trying to figure out what does explain that variation or if it's random. But we tried to find something that would imply that, and we don't see a clear pattern. And that would be really interesting, but we're not sure.
So in the area of Sympatry, how often do you see kind of intermixed populations of Drummondii and Cuspidata?
Yeah, great question. So there's, I can think of 4, but I would say most populations are very close to each other, and fewer populations are actually intermingling. But that's with the caveat that I think, and again, I don't have quantitative data for this, I think Cuspidata is drastically decreasing in frequency. So over the 10 years that I've been going out there, I've seen Cuspidata sites blip out. I think that they're much more susceptible to human disturbance. And so I'm afraid that what we look at now is not what was involved even 15, 20 years ago. So I'm going to keep watching these populations, so maybe over another 10 years I can actually quantify that pattern, but we definitely see sympatric populations. We see more populations neighboring each other, and I think the number are decreasing.
Okay, so we are out of time, but more questions for her in the panel discussion later. So please join me in thanking Robin.